Pre-implantation genetic testing is a technique used to identify genetic defects in embryos created through in vitro fertilization, IVF, before pregnancy. The pre-implantation genetic diagnosis refers to when one or both genetic parents has a known genetic abnormality, and testing is performed of an embryo to determine if it also carries a genetic abnormality. Not all diseases can be tested for in this manner, and single gene disorders are caused through the inheritance of a defective gene. These disorders are classified as recessive or dominant. A recessive disorder requires two bad copies of the gene to pass the disease on to the baby. With a dominant single gene disorder, only one copy of the defective gene is needed to cause the disease. So there are over 1,000 single gene disorders that they have been identified at the present time. Many of these disorders are very rare. However, some common in certain ethnic groups that routine screening to check whether someone is a carrier is recommended prior to getting pregnant. So that is often referred to as carrier genetic testing or screening. So the most common single gene disorders that PGD has been used for are, for example, we have autosomal recessive disorders such as Gaucher disease, Fanconi anemia, adrenal hyperplasia, phenylketonuria, or other common diseases that are used is PGD for autosomal dominant diseases like Huntington's diseases, Marfan syndrome, or some neurofibromatosis. We can test another X-linked disorders due to the X chromosome, like, for example, the fragile X. So that is important for me that in vitro fertilization, the IVF is performed. We have already checked what is the faulty gene or what is the disease that we want to rule out. So the embryos have 12 factorial biopsies done on day 5, on 6, on the blastocyst stage of development. These cells are then sent to the genetics laboratory for evaluation. Then the genetics laboratory does their genetics magic and determines whether the embryos contain that abnormal DNA of the genetic disease in question, in this case. So the testing is done. They use high-tech genetics by the NGS, next generation sequencing. So it analyzes the DNA sequences in the cells for that specific gene in question.